ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.
Hello students, now let us continue with the discussion about factors affecting photosynthesis. So we divided the factors into two categories, external factors and internal factors. So what all will come under external factors? The first one we discussed as light, then we discussed as temperature, carbon dioxide, water, oxygen and mineral elements. But mostly we just coordinated with the carbon dioxide and light. So how carbon dioxide act as a limiting factor, that is left for us to continue our discussion. So before that we will just continue with the internal factors and again we will come back to the carbon dioxide limiting factor. Now if you look at, so we have completed external factors, now let us go with internal factors. Let us go with internal factors. Now if you take the internal factors, which are the internal factors? Now let us start discussing one by one. So first one is nothing but leaf, what you call leaf anatomy. Second one is leaf age. So let us write the list of all factors here. The second one is nothing but leaf age, right? And apart from leaf anatomy and leaf age, we talk about chlorophyll, right? Then other factor is end products, then followed by protoplasmic factors, then lastly we have to discuss about cardinal points. So this all will come under internal factors. So if you take the first factor, what is called as leaf anatomy. So if you take the leaf anatomy part, so whenever in the leaf anatomy if it contains extensive or excess amount of a palisade parenchyma, when there is more content of palisade parenchyma, if more palisade parenchyma is present, where more palisade parenchyma is present? Palisade parenchyma is present in the mesophyll. That means the quantity of palisade parenchyma it's more when compared with the spongy. It's more than spongy parenchyma. It's more than a spongy parenchyma. So here palisade parenchyma it's more than a spongy parenchyma. Then that increases the rate of photosynthesis. Photosynthesis increases. Why? Because we take the proportionate ratio between the palisade parenchyma and spongy parenchyma. Spongy parenchyma has more intercellular spaces and mainly contributes for gaseous exchange. Spongy parenchyma mainly contributes for gaseous exchange because of a large intercellular spaces. Okay, spongy parenchyma mostly contribute for gaseous exchange. You remember transport in plants, the very first chapter? What you have discussed, the transport or gaseous exchange occurs only by the means of diffusion. Diffusion is the only means of gaseous exchange. Diffusion is the only means of gaseous exchange. If you take spongy parenchyma, you find high rate of gaseous exchange within the plant anatomy. If you find more palisade in the leaf anatomy, if it find more palisade parenchyma, that definitely contributes for high rate of photosynthesis. At the same time, you know it very well, mesophyll cells contain more number of stomata, more number of chloroplast, that is highest number of chloroplast. How much means 20 to 40 number of chloroplast is present for each mesophyll cell. This we have discussed in cytology. So, you need to, in every step of lesson when you study, you need to correlate with the first year concept. This is related to cytology. You know, mesophyll contain highest number of chloroplast, that is 20 to 40 number of chloroplast for each cell, right? And this is cytology point. How do you connect with anatomy point? So, you know it very well when you take the leaf anatomy. Let me erase this. When you take the leaf anatomy, what you take here, when you take the leaf anatomy here, right, when you take the leaf anatomy here, what do we say here? Suppose if you take dicot leaf and you take monocot leaf, what you call in case of dicot leaf? In case of dicot leaf, mesophyll is differentiated into both palisade parenchyma and spongy parenchyma. But coming to monocot leaf, undifferentiated mesophyll, undifferentiated mesophyll. Mesophyll is not differentiated. When it is not differentiated, then what you find here? You find parenchymatous cells. When there is more parenchymatous cells, then definitely there is a more rate of photosynthesis. In case of dicot leaf, this is called dorsoventral leaf, because in this case, mesophyll differentiated into palisade and spongy, whereas in dicot leaf it is called isobilateral leaf. What is it called? Isobilateral leaf. Why do you call isobilateral leaf here? Because mesophyll is not differentiated, so you find only parenchymatous cells. Wherever you find more parenchymatous, you can see that there is a high rate of photosynthesis. There will be high rate of photosynthesis. Right? Now let us go with next one, leaf age. Yeah, let us go with next one, leaf age. So, here generally in case of young leaves, rate of photosynthesis is high. Rate of photosynthesis is high or increases, but it becomes in case of older leaves. Older leaves are also called as what? Senescent leaves. Senescent leaves. Senescent leaves means they are nothing but older leaves. In case of older leaves, rate of photosynthesis decreases. Why rate of photosynthesis decreases here? Because of destruction of chlorophyll. Because of destruction of chlorophyll. As chlorophyll get destroyed, destruction of proteins also will get destroyed, because protein synthesis happens within the chloroplast. Chlorophyll pigments will get destroyed. When chlorophyll pigments are destroyed, when proteins are destroyed, then in older leaves the rate of photosynthesis decreases. Leaf age also becomes one of the internal factor contributes for rate of photosynthesis. Right? Now, let us take the next one as chlorophyll pigment. Chlorophyll. Where chlorophyll occurs? Chlorophyll occurs within the chloroplast. Chlorophyll occurs within the chloroplast. So, when you take the chlorophyll occurs within the chloroplast, what are very important things here? So, very importantly you know that chlorophyll A is the main pigment or major pigment, right, which plays a very important role, a major pigment. Then we also find what are called chlorophyll B, chlorophyll C, D and E, right, along with other pigments like carotenoids and xanthophylls. And these carotenoids and xanthophylls, these all act as, what do we call them? These all are called as accessory pigments. What do they do here? These all accessory pigments, what is the main function here? They protect chlorophyll A. They protect what? Protects chlorophyll A from solarization, or we call photooxidation. What is meant by photooxidation? Quite a number of times we are discussing, it is nothing but destruction of chlorophyll A. Because chlorophyll A is nothing but the major pigment we find, it is called universal chief pigment or most central pigment, very important for rate of photosynthesis. Now you know at this point of time in plant kingdom you have rhodophysi, phaeophysi, chlorophysi. Where do you find chlorophyll A and B, A and C, A and D? This all you have to need to recollect it once again. Next, apart from this, here when you take chlorophyll mutants are called albinos. Very important. Chlorophyll mutants are called as albinos. So, what are chlorophyll mutants here? So, chlorophyll synthesis may not happen properly. At the time of chlorophyll synthesis, because of the mutations, chlorophyll structure is not completely formed. So, there is going to be impact on the structure of chlorophyll get affected because of this mutants. There is no complete structure of the chlorophyll, and such kind of chlorophyll mutants which are formed without proper structure of chlorophyll, they are called as what? Albinos. So, what do we say? Formation of chlorophyll mutants, meaning formation of albinos, they make decrease of photosynthesis, or you can write directly decreases photosynthesis. Okay. So, what do we say here? Formation of albinos decreases photosynthesis. Generally, this is also been studied and observed by the scientist. Try to remember Emerson. Emerson is the one who studied about albinos. So, what are the contributions of Emerson? Red drop and photosystem 2 and 1, right, and even in bacteria 890 nanometers beyond that what you call photosystem 1 in bacteria. It studied about how the photosynthesis happens in bacteria. Similarly, the same Emerson person, Emerson enhancement we studied, red drop we studied, red fall we studied. Same person also observed and identified albinos. If albinos are there, then that decreases the rate of photosynthesis. Right. Now, let us continue with the next factor. Now, we say end products. How do you think end products inhibit photosynthesis? In some cases what happens? End products inhibits the process of photosynthesis. Guys, this we have studied in enzymes. In enzymes at the time when we take what is the types of inhibition when we studied, then we studied an important concept called as a feedback inhibition. We have studied what is called as feedback inhibition. What is feedback inhibition? So, it starts with the substrate, let us take A. A produces intermediates like B, B produces C, C produces D. Let us imagine E is nothing but an end product. What is the end product produced? Let us take it is nothing but glucose or fructose, or you can take sucrose. Right. So here when you say here A is giving B, B is giving C, C is giving D, and these all are nothing but what? There are so many intermediates. There are so many intermediates. And what is A? A is nothing but the first substrate. Right. And E is nothing but the product. In between you can see there are so many intermediates. So generally in a couple of reactions where A is giving B, C, D, there are so many intermediates, finally we are getting what is called as product. Now sometimes what happens, if all mesophyll cells are filled with the glucose, then no more space to accommodate the glucose, then the end product glucose itself will go and inhibit the enzyme of the first reaction. So this is what is called as feedback inhibition. So what happens if all mesophyll cells are fully filled, then there is no space to accumulate more glucose. So photosynthesis has to get stopped. Who will take the responsibility? End product. End product E will go on end product. I am just writing here. End product controls the enzyme of first reaction. End product go and control the enzyme of first reaction. When this enzyme is blocked, when this enzyme is stopped working, when enzyme is totally blocked, then the conversion from A to B will not happen. Then it stops the reaction, then no more synthesis of glucose. Until glucose utilized for various metabolic activities, they will not allow the reaction to happen. So end product itself will go and control the enzyme of the first reaction, so that reaction stops. It's called as what? Feedback inhibition. Right. Similarly, let us talk about protoplasmic factors. What we have is protoplasmic factors. What are protoplasmic factors we have? The protoplasm plays very important role for making the cell to be hydrated with water, then only photosynthesis takes place. So here whenever the water content is more, so water concentration is more, where within the protoplasm, if the water concentration is more within the protoplasm, then rate of photosynthesis increases. Rate of photosynthesis increases. Suppose if there is water concentration decreases, if water concentration decreases, then that may lead to dehydration of the protoplast. So now you can see dehydration, or you can say dehydration, yeah, dehydration of protoplast, meaning protoplast will become totally dry and may not support the process of photosynthesis. So dehydration of protoplast itself, it decreases the rate of photosynthesis. So increase in photosynthesis and decrease in photosynthesis also depends upon the sensitive factor called protoplasm, living matter. If the cell has sufficient amount of water, then only the photosynthesis takes place. Of course, sometimes the plant have plenty amount of water, but cell may not get so much of water. Then it will be dehydrated, then rate of photosynthesis, even though other conditions are favorable, but there is a decrease in rate of photosynthesis. So sometimes protoplast also act as a factor, it's called protoplasmic factor. What happens sometimes when a new plant, we are just getting it from some other environment, we are trying to plant in our own home or in our home. What happens here, the new plant was very healthy from the place where you brought. As soon as you keep it within the soil, in soil all conditions are favorable, a new plant you have planted which you brought from some other place, where light is available, temperature is available, carbon dioxide is available, you are pouring plenty of water, but still the plant will undergo the process called as wilting. Why? All leaves will become very dry. Why all leaves becomes very dry? It's because of evaporation of water. Whatever water it takes from the soil, total water will get evaporated because protoplasm may not have sufficient amount of water. As a result also there may be decrease in rate of photosynthesis, then leaves becomes very dry. It takes some time for the plant to get adjusted to the new environment. At that time maybe it may be the reason of protoplasmic factor. Right. Now let us come to the next one, cardinal points. What is the next one? Cardinal points. Now what are these cardinal points? Cardinal point, it may represent, talk about what are called as amount or what is called concentration or amount of factors which are responsible for rate of photosynthesis. What do you say? What generally we say? What are cardinal points? The concentration or amount of factors which are generally responsible for rate of photosynthesis. How much quantity is there? What is that factor responsible and what is the rate of photosynthesis? That can be represented with the help of what are called as cardinal points. Now we have three cardinal points. If you take, we say generally we say a minimum cardinal point. What is a minimal cardinal point? Minimum cardinal point means below the concentration of this, below minimum concentration, photosynthesis does not take place. Photosynthesis does not take place. Okay. So what is minimum cardinal point? The minimum concentration below which photosynthesis does not take place. It will not start at all. The process of photosynthesis will not start at all. So below minimum concentration photosynthesis does not take place. Then the next one we have is nothing but optimum cardinal point. Optimum cardinal point. What is optimum cardinal point? We say now it's a point at which the rate of reaction occurs at high velocity. What's the meaning of this? Optimum cardinal point meaning the point at which rate of reaction occurs with high velocity. Meaning here suppose if you talk in terms of maximum, in terms of photosynthesis, a high or you can take a maximum photosynthesis takes place. PS means photosynthesis takes place whenever there is an optimum concentration of a particular factor. Optimum concentration of pH, optimum concentration of carbon dioxide, optimum concentration of water means maximum water is there, maximum carbon dioxide is there, then the rate of reaction occurs at a faster rate. So maximum rate of reaction occurs with the highest speed of velocity. That means it is recorded with high photosynthesis or maximum photosynthesis. If you talk about that process is nothing but a photosynthesis. Similarly, then the next one is referred as maximum cardinal point. So let us write with blue with this. So all two I am writing with that. Maximum cardinal point, maximum cardinal point. What is this maximum cardinal point? Says that means above this, the point above this, the point above this, the process stops. The process stops. The process stops. The point above this, the process stops. Meaning this is a concentration beyond which the process stops. That means when you say that, so in terms of photosynthesis, beyond optimum, so here the photosynthesis, PS means photosynthesis completely it will get stops. Beyond optimum range, photosynthesis completely it will get stopped. So when photosynthesis is stopped, then photosynthesis rate becomes constant, then at that time what you say, the process stops completely and photosynthetic yield decreases. So we have cardinal points. What are we have cardinal points? Minimum cardinal point, optimum cardinal point, and we have maximum cardinal point. What is minimum cardinal point? Below minimum concentration, photosynthesis does not take place. Photosynthesis value becomes 0. It will not start. Why? Because required quantity of the factor is very minimum, very less. What is optimum cardinal point? The point at which the rate of reaction occurs at a high velocity. Meaning what here? Rate of reaction takes place at a high rate. Maximum rate of photosynthesis happens. That is called as optimum cardinal point. Maximum cardinal point means what? The point above this, the process stops. Let us apply this for study of carbon dioxide. So what we are going to study now? Internal factor we have carbon dioxide is there. So when you take the carbon dioxide in terms, what is that we have to study? A black man's limiting factor. What is that we are going to study? A black man's limiting factor. Regarding carbon dioxide, we are going to study black man's limiting factor. Now this black man's limiting factor, it's purely based on a Liebig's limiting factor. So before Blackman, already there is another scientist who has given about something about limiting factors. So when you talk about limiting factor here, already Liebig has studied. Based on that, Blackman has given a complete study of limiting factor in terms of carbon dioxide. So what we are going to discuss now? Carbon dioxide. What we are discussing? A Blackman's limiting factor. Now we are discussing Blackman's limiting factor. Just remember a point based on Liebig's limiting factor. Now if you look at this Blackman's limiting factor, if you try to remember this Blackman's limiting factor. Now according to Blackman, when a process is conditioned by its rapidity, then the process controlled by number of factors and space by the lowest factor What is the meaning of this? This is a statement according to Blackman's. According to Blackman, when a process is conditioned by its rapidity, rapidity means so frequently the process is taking place. So, when process is taking place so frequently, the process is controlled by a number of factors. There are so many factors. Rapidly means it keep continuously taking place. When a process is conditioned by its rapidity, the process is most essential process. That means we are speaking about photosynthesis. It will occur continuously throughout the day. Rapidity means it is happening one after the other. Now, then the space by the lowest factor, then it will be completed by the, then it will be stopped or inhibited, or if it is more affected by the one of the lowest factor, or you can also write one of the slowest factor. Any one factor will make the photosynthesis to stop after certain period of time, and that factor will act as, that factor itself act as, we call that as a limiting factor. We call that factor as what? A limiting factor. Now, let us study about this part here. For example, we take light versus carbon dioxide, and along with that we are taking rate of photosynthesis. Rate of photosynthesis. Then here we are taking carbon dioxide and simultaneously also taking with the interaction with the light. I told you, no, for the last class, carbon dioxide and light together, they play a very important role in controlling the rate of photosynthesis. So, when you take here, initially carbon dioxide concentration is 0, let us take A. So, below A there is no photosynthesis. Then when carbon dioxide concentration increases, slowly rate of photosynthesis also increases, then it becomes constant. So, B and C, that means here with carbon dioxide concentration photosynthesis increased. So, here photosynthesis increased, PS means photosynthesis increased, and then carbon dioxide concentration or rate of photosynthesis. So, here you can write photosynthesis became constant. B to C represents photosynthesis became constant. Then further rise here 0.03, 0.5. So, further rise of carbon dioxide concentration, then rate of photosynthesis also increased and then become constant. Let us take D and E. Simultaneously with we are also rising light is also getting increases. At high carbon dioxide concentration, now photosynthesis has become stable. Further there is increase in light. So, there is increase in rate of photosynthesis, this time with the effect of light. Let us take light intensity 1, means light has increased up to certain level, light intensity 1. Then rate of photosynthesis also increased from D to F. So, here we say that D to E, then F to G. Then there is further rise in light intensity, then again photosynthesis becomes constant. That means here with the increase in carbon dioxide concentration, photosynthesis increased up to certain level, then it became optimum. Beyond that, if still light is increased, again it is further increased, again photosynthesis became constant. Now, this is what is being explained by Blackmans. What's the meaning of this? When a process is conditioned by its rapidity, the process is controlled by a number of factors or spaced by the slowest factor. That factor itself is called as limiting factor. What is the meaning of that? What is the meaning of this limiting factor? We say limiting factor means, suppose carbon dioxide is favorable, carbon dioxide concentration is favorable, then water is there for photosynthesis to happen, oxygen is there, mineral elements is there, but light is not there. Then light act as limiting factor, because remaining all conditions are favorable, then light act as a limiting factor. It stops the photosynthesis, or we say photosynthesis got stopped even though other conditions are favorable, but photosynthesis is inhibited because of light, then it is called as a limiting factor. Other conditions are favorable, but only light is making the photosynthesis to decrease. At that time, the light will act as a limiting factor. Suppose even light is also available, water is available, mineral elements are available, but carbon dioxide concentration is less. less carbon dioxide concentration is there, then carbon dioxide stops the process called photosynthesis. At that time, carbon dioxide acts as a limiting factor. The factor which inhibits rate of photosynthesis itself is called as limiting factor. Why? Why any one of the factor act as the slowest factor or limiting factor? If any one factor is not controlled, then what happens? Rate of photosynthesis is keep increasing, keep increasing, keep increasing. More food is produced. There may not be space to accommodate that food. If all mesophyll cells are fully filled, there won't be any space to store that particular food. Right. So, it should be controlled at one point. Any one of the factor will take up the responsibility to control. That factor act as a limiting factor. Now, what do we say here? The minimal factor. Cardinal points are how many? Minimum cardinal point, optimum cardinal point and maximum cardinal point. Let us study about this. Look at the graph here. A. At A, carbon dioxide concentration is 0. So, it is 0. Then there is no photosynthesis. There is no photosynthesis. At A, carbon dioxide concentration is 0, then no photosynthesis. The point below which there is no process takes place, it is called minimum cardinal point. Then optimum cardinal point, that means let us take that carbon dioxide concentration increased from 0 to 0.5. Then what happened? Photosynthesis also increases from A to B. Okay, if the carbon dioxide concentration increased from 0.03, 0.5, then rate of photosynthesis also increased from A to B. Rate of photosynthesis increased from A to B. Right. Then if the carbon dioxide concentration increased from 0.05 to 1 milligram, then after that it became constant. B2C it became constant. After some time photosynthesis became constant. Then again the carbon dioxide concentration increases from 0.05 to 1 milligram. Then again photosynthesis also increases from where to where? D to E. I mean B to E. B to E. So from B to E, B to D here, from here to here what? 0.03, 0.05. What? Carbon dioxide. So up to A to B increased, B to C constant. Again it increased up to 1 milligram. Then rate of photosynthesis increased from where to where? B to D. So here you take it as B to D. Right. And beyond 1 milligram, then rate of photosynthesis become constant. Rate of photosynthesis becomes constant. Then at that time what is supplied here? Then light is supplied. Light is supplied. When light is supplied here, again rate of photosynthesis increases from where to where? D to F. Increases from D to F. If light is supplied, then rate of photosynthesis increases from D to F. So here light is added. When light intensity added here, even though carbon dioxide concentration become maximum, when light is added, there is an increase in rate of photosynthesis from D to F. So that is nothing but optimum. Then beyond carbon dioxide concentration, beyond light, then what happens? Beyond optimum light, then what happens? Rate of photosynthesis constant, or rate of photosynthesis will become decreases. Then that is called as what? That is called as what? Maximum. What is maximum here? Beyond carbon dioxide concentration, beyond optimum light, meaning beyond high carbon dioxide concentration, rate of photosynthesis stops. That is maximum volume, or beyond optimum light also rate of photosynthesis becomes constant. Constant from where to where? F to G. F to G is rate of photosynthesis becomes constant. So these are cardinal points, and that explains that carbon dioxide will stop the rate of photosynthesis. Carbon dioxide acts as a limiting factor, that is explained by a Blackman's limiting factor. Blackman limiting factor, the definition is important. When a process is conditioned by its rapidity, the process controlled by number of factor, space by the lowest factor or itself is called as the limiting factor. Accordingly, in this case carbon dioxide acts as a limiting factor. In minute science.